Welcome back to Breakdown of Science. I'm Jan.
And I'm Michelle. Today we have a very complex topic. Okay, not a very complex topic, but us to discuss. So we're diving deep into the world of gene modification and gene editing. So this topic spans decades of science, touches on every branch of life from microbes to humans, and also raises profound ethical questions. So yeah, buckle up.
Yeah, absolutely. And we'll cover where the idea first emerged, how the experiments began, the various methods scientists have used over the time, and especially CRISPR, the tool that set the gene editing world abuzz, on fire in the last decade. And we'll also talk about what's possible now, some of the real world applications, and what the future might hold, along with, as we said, the ethical and social debates and discussions that are currently ongoing in the regards of gene editing.
And since this topic is, big, we will actually be dividing this episode into two parts, with the second part being uploaded next week. And since the topic is very technical, we will also do our best to, explain the terms and the topics as simple as possible.
Yep, and so let's just get started and go into Chapter 1: The History of Gene Modification and Editing.
Yeah, so to understand gene editing today, we need to go back further than you may think. Humans have been influencing, the traits of plants and animals for thousands of years through selective breeding, as in like picking individuals with desirable characteristics and breeding them. But that was just based on choosing all organisms, but not directly changing the DNA.
Yep, exactly. And so the conceptual leap to changing the actual DNA began with some basic genetics back in 1866 with Gregor Mendel, who you might have heard back in your biology class in middle school or high school, who was actually a pastor at a church and just grew pea plants in his greenhouse and discovered this. He published this small experiment that he did with his pea plants and showed how certain traits are inherited, which laid the groundworks for genetics. But the real molecular breakthrough came in 1953 when Watson and Crick described the double helix structure of DNA. So that's the twisted ladder shape of the molecule that carries the genetic instructions. But one thing that I will have to clarify is that it was Rosalind Franklin and Morris Watkins who were the ones that actually saw the structure of DNA first in the same year, and on whose work Watson and Crick based their description on.
So once scientists knew DNA structure, they could imagine manipulating it. And fast forward to 1972, Paul Bug and his colleagues made the first recombinant DNA molecules. Recombinant DNA means, joining DNA from two different sources. So basically, cutting and pasting pieces of genetic code. And this was revolutionary because until then, transferring genes between organisms at the molecular level wasn't really that possible.
Yep, and that started then the safety concerns, the ethical concerns. Could this create dangerous organisms? So
Mm.
in 1975, scientists met at the Aslimor conference to set certain guidelines for recombinant DNA work. They agreed on safety measures. This was one of the and they agreed on safety measures there. This was one of the first times scientists self-regulated on potential biohazards, especially at this scale.
And then in the late 1970s and early 1980s, the recombinant DNA started yielding tangible products. So, for instance, in 1982, genetically engineered insulin, which is made by inserting the human insulin gene into bacteria, became the first biotech-derived medicine approved for treating diabetes, and that demonstrated the real medical benefits.
Yep. And meanwhile, researchers were tinkering with animals, and in 1974, Rudolf Jenich and Beatrice, Beatrice,
Actress?
Beatrice, sorry,
Yeah, no worries.
Beatrice Mintz injected a viral gene into mouse embryos and showed it passed into offspring. And that was the first demonstration of a transgenic animal. And transgenic just means an organism carrying a gene from another species, with just trans meaning different, genetic being genes, so just a different gene. Anyway, by around 1980s, labs produced mice with reporter genes. For example, the jellyfish green fluorescent proteins that's used a lot, and that glow under certain light to track cell behavior.
And all these early steps, the recombinant DNA, transgenics, first gene therapies, they built the foundation. Because in 1990, the first gene therapy in human took place, which is treating a child with ADASCID, which is basically a severe immune deficiency, by inserting a working gene into their cells outside the body and then putting them back. And that was, as we all know, a first glimpse of treating disease by changing genes.
Yeah.
Yeah.
And so by 1990s, gene modification was real. We could add new genes or disable genes in cells or animals, but the tools for making precise targeted changes, as in like changing one specific spot in the genome, were limited. And that led to the development of engineered nucleases, which are basically molecular scissors, later on, which we'll also cover later on. But first, let's clear up some definitions and classifications that are used in genetic engineering in chapter 2 with definitions and classifications. Very original, very creative.
Yeah. So people often mix these up. So gene modification versus gene editing, they're similar but not exactly the
Yeah,
same thing.
yeah, exactly. And we've also like mixed it up quite a few times because in
Yeah.
most cases people use it interchangeably and
Mm.
in general have this similar sort of idea of like it's doing some sort of change at the gene.
Yeah.
But so what specifically it is? What is it specifically? So gene modification or genetic engineering is kind of an umbrella term. It
Mm.
usually means changing an organism's DNA in a big way, like inserting or deleting entire chunks of DNA. Sometimes it even involves bringing in genes from another species, as we said with the transgenic mice,
Mm.
and or like when scientists add a bacterial gene to corn so it can resist pests. That's genetic modification.
Exactly. And to make it simple, it's like copying a recipe from one cookbook and then pasting it into another one. And suddenly your grandma's cookbook has a sushi recipe. Meanwhile, gene editing is definitely more precise, I would say. So instead of copying recipes from other books, you're just going into an existing recipe and tweaking it and maybe changing an ingredient or just fixing a typo.
Yeah. So for in that analogy, it's like replacing 2 cups of salt with 2 teaspoons. You're working with what's already there, but making it better or just different. And now within gene editing, there are a few techniques that you all should know. And the first up is knockout.
Yeah, so that's when you turn a gene off, basically. So scientists mess with it just enough, like usually by adding or deleting a few letters, nuclear bases actually, so it stops working, like crossing out a step in recipe, or you just skip, so that you skip that ingredient altogether.
Yeah.
Yeah.
And then there's knock in, which is more or less the opposite. You're inserting something new into a specific spot, like replacing a broken gene with a working one. Or, following the analogy that we're going with, swapping out a messed up sentence in your recipe for one that's actually make sense. Like maybe the terminology from way back then changed, and now you have to change the wording to understand that this is the food it's talking about, or this is the measurement it's talking about, or this is the process it's talking about.
Exactly. And we can also not forget about the transgenic organisms. So that's when you take a gene from a totally different species and add it in. This used to be the main method for giving organisms new traits, and that's basically like adding a recipe from Thai cuisine into Italian cookbook. If we are still using the same analogy, yeah.
Yeah. And now here's where it gets even more interesting, actually. Whether the edits are somatic or germline. And we'll discuss these in a second. So, somatic means body cells, like the cells in your liver, blood, skin, all those. If you edit those, the changes only affect the person being treated. So only you. It doesn't get passed on to your kids. And so it's like editing your copy of the cookbook and no one else's copy changes.
Exactly. But germline editing, that's when the changes are made to, the eggs, the sperm, or the embryos. So the edits are inherited by future generations, and that's like changing the original family cookbook that gets handed down to everyone, and now everybody's recipe includes a little tweak. Yeah.
And most countries are okay with somatic editing, of course, under very tight rules. But germline editing, that's a lot more controversial. It's either banned or super restricted in most places. And so the ethics get pretty complicated fast.
And in the last few years, CRISPR has been, the up and coming genetic editing tool that is being used and researched in many labs. But what were the tools that came before? And now we'll move on to chapter three, but the early gene editing tools before CRISPR.
Yeah. So, all right. Before CRISPR took the spotlight, scientists were already crafting molecular scissors that could cut DNA at specific spots. Two big players back then were zinc finger nuclease, or SFNs, and talents, TALENs, which stands for transcription activator like effector nucleases. Quite a mouthful.
Yeah, exactly. So let's unpack those names. So a zinc finger is, in a nutshell, yeah, stabilized by a zinc ion, and each finger latches onto about these three DNA base pairs. And fusing several zinc fingers together lets you recognize longer DNA sequences. And then you attach a nuclease, which is an enzyme that cuts DNA and RNA like a blade, called FOK1. Focal? Yeah, okay. Focal to cut the DNA once two of these complexes bind on, the both, no, on opposite strands. Yeah.
And hence, SFN, or zinc finger nuclease, that's
Yeah.
the name. That's why it's called that.
Yeah.
So, two halves of a cutter have to come together before you get a cut. Like two people each holding half a pair of scissors, finding their marks on a rope, and then snipping it. The catch, though, is that every new DNA target needs a brand new set of these zinc fingers, and designing them was slow and expensive.
And enter talents around 2010, which is transcription activator like effector nucleases. So,
Yeah, say
transcription, what is
that 5
that?
times fast.
Yeah. So, what is transcription? So, that's basically the process by which DNA is copied into RNA. Activator like they are just proteins that resemble activators. They are molecules that bind the DNA and boost transcription of nearby genes. And the effectors are basically just agents that bring out a specific effect, in this case, the ability to bind a targeted DNA sequence. So there you have it, transcription activator like effectors. So they're just DNA binding proteins modeled on bacterial factors that hijack the plant transcription. So
Mm-hmm.
scientists take those tail repeats, each recognizing one DNA base, string them together to match their target, and then fuse it to a focal nucleus, which is the cutting blade. Yeah.
And it's like snapping together Lego bricks. Each
Mm,
brick reads one letter of the recipe, then attaching scissors so you can cut precisely where you've spelled your sequence.
mm,
It's bulkier than ZFNs, but way more modular.
exactly. And despite their quirks, ZFNs or ZFNs,
Yeah.
doesn't matter. And talents proved precise genome cutting was possible, and they definitely enabled everything from gene knockouts to targeted gene replacements, and even reached early clinical trials like using ZFNs to make HIV resistant blood cells. Yeah.
But the big drawback, and also one of the reasons we're not using it today as much, is that for each new gene target, you had to design and build entirely new proteins.
Mm,
But that's why CRISPR, where you only swap out a small guide RNA, was such a revolutionary leap in simplicity and scalability. And that leads us to our next chapter, CRISPR-Cas systems, the revolution.
ooh.
Uh, so, okay, now let's talk about the CRISPR. It's short for Clustered Regulatory Interspace Short Palindromic Repeats. If talent was a mouthful, I don't know what this was, but it indeed is that. But
Yeah.
here's the gist basically. So, bacteria use these repeated sequences as part of their immune system, or what would resemble an immune system, snipping out snippets of viral DNA and tucking them between those repeats so they quote unquote remember past infections. So that when they get infected again with that virus, they say, oh, okay, I recognize this. This is an infection. Let's fight it.
Mm, yeah, exactly. And yeah, in 2012, Jennifer Doudna and Emmanuel Carpentier. I
Carpentier.
have no idea. I'm so sorry if I butchered the name. Anyways, they showed that you could hijack the system in a test tube, and they found out that the Cas9 protein, guided by two RNAs, which is the CRISPR RNA and the trans-activating CRISPR RNA, they could cut any matching DNA sequence that you wanted. Yeah.
And the real game changer of this was that they fused those two RNAs into one. And
Mm,
it's what we now call the single guide RNA, or in short, the sgRNA.
yeah.
So now instead of juggling two pieces, you've got one guide RNA that tells the Cas9 protein exactly where to cut. You can think of Cas9 as one of the molecular scissors we've been talking about, and the sgRNA as the address label stuck on the DNA saying cut here, like a tape around the rope to indicate it.
Exactly. And soon after, in early 2018, labs led by Feng Shang, George Church, and others adapted CRISPR-Cas9 to work inside living human and mouse cells, so showing you could get those precise cuts on more complex cells. And that definitely kicked off the flood of CRISPR research we see today.
Mhm.
So now let's break down on how CRISPR-Cas9 works. So as Jan mentioned before, there's the single guide RNA. It's exactly not exactly, but it's like about 20 RNA nuclear bases long, which matches the DNA that you want to cut. So you can think of it as a post-it with the exact address of where to snip, as Jan mentioned before. And then there is the PAM, that stands for Proto Spacer Adjacent Motif. So it's a short DNA motif next to the target, which is usually NGG for the common Cas9. So N is just any nucleobase, and G stands for guanine, which is one of the molecules that make up the DNA. And Cas9 won't cut unless it sees that PAM right beside your target. So basically, it's kind of like a street light turning green and letting it go and cutting. Yeah, and then there's Cas9 protein. It has two nucleus plates. The HN domain cuts the DNA strand that matches the guide, and the domain is kind of like a sequence of amino acids, which are the building blocks of the proteins. And then there's also the RU. the roof C-like domain that cuts the opposite strand. And the RUVC-like domain is a protein that resembles one found in bacteria that helps in cutting. So once the single guide RNA and Cas9 find the right spot next to the PAM, the Cas9 makes a double-strand break, so snipping both strands of the DNA helix about, 3 bases upstream of the PAM. You guys can think of it like a scissor slicing both sides of a rope at exactly the right spot.
And
Yeah.
by upstream, it means as in 3 base pairs in front, 3
Yeah.
base pairs after the PAM sequence.
Yeah, thanks.
So, once cut, it comes with the cellular repair, and there is 2 methods that are done. So one is the non-homologous end joining, or NHEJ. The cell quickly ties the ends back together, and this often causes a mixing of a few of the letters by either adding or removing, which can end up either knocking out genes or even knocking in. And this basically is like tying the rope back hastily after cutting, so you might end up losing some of the fibers or even end up with a big knot. And then there's also homologous homology-directed repair, or HDR. This is where if you would supply a DNA patch with edges that match the cut site, the cell can use it to repair the break more precisely. And it's like fixing a hole in a piece of fabric by sewing in a matching patch, rather than just pinching the torn edges together, giving you a smooth, cleaner repair. And I do want to say that scientists rarely are able to create good-sounding acronyms, and that's something that we just have to accept at this point, honestly.
Yeah. So while the HDR is great, it's also sadly inefficient, especially in dividing cells like neurons, heart muscle cells, or retinal cells, blah, blah, blah. And that's why scientists have been inventing the base editors and prime editors to make the precise tweaks without relying on HDR. So yeah, Jan will talk about other CRISPR systems.
Yes, of course, yeah. So there isn't just... So CRISPR doesn't just stop at Cas9. There are other systems as well, and one is Cas12, which is also known as CPF1. So like Cas9, it cuts DNA, but instead of making a clean, straight cut, it creates a staggered or sticky end, basically like a step, which can be useful for certain types of genetic engineering. And Cas12 also reads a different PAM sequence. It looks for something like TTTV instead of the GG that we mentioned for Cas9. And here's a cool thing. Cas12 can process multiple guide RNAs from one long transcript all by itself. So imagine scissors that leave a little fringe when they cut the rope and only work when they see a different kind of street sign, for example.
Yeah, and then there's Cas13, which is kind of a different beast. So it doesn't cut DNA like at all. It targets the RNA, the messages that cells send to make proteins. And once Cas13 finds the exact RNA that it's looking for, it doesn't just stop there. It goes on, on a shredding spree, I suppose, cutting up other RNAs nearby too as well. So scientists have actually harnessed this collateral cleavage ability to develop powerful diagnostic tools. So one of the most famous ones is called Sherlock, and yes, it is a bit of a link to Sherlock Holmes, the detective, but it's also an acronym, so it stands for Specific High Sensitivity Enzymatic Reporter Unlocking. No, that's a mouthful. Anyways,
again, there's, when it comes to acronyms, it's one of two ways. Either you're just mushing up the first letter of all the words, and leaving it that, or if they can make a pun out of it, then they put effort in specifically choosing the specific words to create it, as they have done with Sherlock and many more.
Yeah.
Sometimes it's easier, sometimes it's not.
But I definitely like the Sherlock one. It's just too much to think about.
They cause a bit of fun,
Yeah.
but it's that usually when it comes with a pun acronym, then you
Yeah.
have to understand a bit more specific understanding, like what it actually means, rather than
Mm.
its own letters. But that's a
Yeah.
discussion for another day.
Yeah. Definitely.
So, uh, this Sherlock method, it uses the Cas13's ability to just shred nearby RNA as a signal. If the target RNA, say from a virus, is present, Cas13 activates and starts cutting up reporter molecules. Those release a signal, like a glow or a color change. So basically, it tells you whether something specific is there, like viral DNA in a COVID test. And you can think of it like scissors meant for cutting messages, the RNA, rather than the DNA master blueprint. And once it finds the message it was looking for, it freaks out and shreds all the messages in that area, which in this case is a good thing because we can see that chaos as a signal that the target was found.
And obviously, CRISPR has also evolved beyond just cutting the DNA. So, one cool example would be the base editors. So, these are just made by fusing a modified Cas9 called nicase, because because it only cuts one strand of DNA. But a special enzyme that can chemically change one DNA nucleobase into another. So, instead of making a full break in the DNA, you're just flipping, let's say, a C to a T or an A to a G. Yeah.
And then there are prime editors, which take it a step further. They also use the Cas9 nicase, but this time it's combined with a reverse transcriptase, an enzyme that can write new DNA based on the RNA template. And the guide RNA in this case is special. It not only tells Cas9 where to cut, but it also carries the blueprint for the edit you want. So the reverse transcriptase then uses that blueprint to write the new sequence at that nick, at that position. And these next gen tools aim to cut down unwanted side effects, like off-target cuts or random insertions, and boost how often you get the precise edit that you want. And now we've talked about how these edits work and stuff, but how are these proteins, these nucleases, these enzymes actually delivered inside of it, inside the cells? And that's our next chapter. Chapter 5, the delivery methods.
Yeah, so once you have your CRISPR tools, the Cas proteins, and the guide RNAs, the next step is actually getting them into the cells so you want to edit. And that's not always easy because the delivery method really depends on the target. Like, is it in the body or in a dish, dividing or not, and what kind of tissue are we talking about? So first, let's start with viral vectors, which are kind of like biological delivery trucks. So one of the most common one is AAV, or adeno-associated virus. So it's often used in gene therapy because it can carry a Cas gene and a guide RNA into tissues like the retina. And the AAV doesn't usually integrate into the genome. It stays separate in the cell, which makes it safer in some contexts. Yeah.
Yeah. And there's also the lentivirus, which is a type of retrovirus. And this one actually integrates into the genome. So it's often used for what's called ex vivo editing, like editing blood cells outside the body before putting them back in.
Yeah.
And both have their pros and cons. The viral methods are usually pretty efficient at getting into cells, but AAV has limited cargo space, so you can send less information through it. And lentivirus can come with its own risks, like triggering an immune response or altering the genome in unexpected ways.
Yeah, and when there's viral methods, there are definitely non-viral methods. So they're just especially useful when you want to avoid these kind of risks. So one is lipid nanoparticles, or in short, LNPs. So they are just tiny fat bubbles that you can, visualize. that carry the mRNA for Cas9 and the guide RNAs. So they have been used for editing liver cells successfully and are the same kind of tech used on mRNA vaccines.
And another method is also electroportation. Electric teleportation, one way you can say it, where you zap cells with an electric pulse to open little pores in the membrane, so in the surface of the cell. That lets the Cas9 protein and guide RNA slip in, and it's mainly used for editing cells in the lab, like T cells, which are one of your immune cells. And then there's also the RNP delivery, where you pre-assemble the Cas9 protein with its guide RNA before sending it into the cell. This method works fast and breaks down quickly, which helps minimize off-target cuts.
Yeah,
so it's more specific.
Yeah, definitely. And finally, for the real targeted applications, like editing embryos or certain tissues, you can use the physical injection. So, for example, scientists can microinject their CRISPR tools directly into a fertilized egg or even a specific tissue. So, just directly, injecting the tools exactly where they are needed.
Yep. And so, in the end, the best delivery method depends on a bunch of factors. What kind of cells you're targeting, whether they're dividing or not, if you're editing inside the body, which is called in vivo, or outside, and reintroducing the cells later, which is, as we said, ex vivo, and how safe or efficient you need the process to be.
Yeah. And as we said at the beginning, this is a big topic, and today we've only gone into the moderate depth, and there's definitely still a lot more to unpack.
There's quite a lot more.
Yeah.
More or less, another half of this could end up being in the next part. So next time, we'll continue this journey by diving a bit deeper into how gene editing actually works. We'll talk about its applications more thoroughly and go even through some real-world case studies and explore the ethical questions and controversies that come with this powerful technology. We've already mentioned that it does raise ethical questions, and we've touched on them very briefly, but next time, we'll go. Yeah, exactly.
And until then, feel free to share your thoughts or questions in the comments and pass this episode along to anyone curious about gene modification and editing. Also, make sure to follow us on Spotify, YouTube, and Instagram so you know when the part 2 drops. So for now, it's goodbye, and we'll see you in part 2.
Yeah, goodbye.
Bye.